Very few events in the biology of microorganisms have become as predictable as the regular re-emergence of influenza outbreaks. Based on antigenic differences, influenza viruses were classified into three groups, A, B, and C. Their RNA genome is segmented into 8 segments for type A and B viruses and 7 segments for type C viruses. 2 of these genes encode hemagglutinin and neuraminidase, essential proteins required for infectivity. Hemagglutinin is responsible for the viral attachment to host cell receptors, and neuraminidase facilitates the dissemination of the virus within the organism. 2 features make influenza viruses become particularly major public health challenges. One of them, the lack of proofreading, is a general characteristic of RNA viruses. As a result of high error rates during viral replication, mutations are constantly introduced into the viral genome and make the resulting viruses slightly different from one season to the next. This process, known as antigenic drift, occurs in type A, B, and C viruses and explains why people who previously had the flu may become reinfected. In addition, the segmented organization of the viral genome facilitates the exchange of genetic information between 2 strains that are co-infecting the same organism. This enables the emergence of reassortant viruses. This process, known as antigenic shift, was described only for type A viruses and results in major antigenic changes. 2 parental viruses can generate 256 distinct combinations by this process, providing a very powerful mechanism for genetic diversity. Based on these 2 sources of variation, 2 types of influenza infection were described: seasonal and pandemic. Seasonal influenza occurs annually in a very predictable way, usually in winter in temperate regions and throughout the year in tropical regions, although seasonality is not very well understood. Pandemic influenza emerges once every few decades. For example, 3 pandemics were described during the 20th century. Most recently, the emergence in April 2009 of a new reassortant H1N1 virus marked the first influenza pandemic of the 21st century. There are certain major differences between seasonal and pandemic influenza. Little or no immunity exists to pandemic viruses, healthy people are at very high risk, and there is a high and often unpredictable mortality toll. Additionally, vaccines in pandemic outbreaks usually become available very late during the outbreak. Importantly, while the risk groups for seasonal and pandemic influenza are different, several segments of the population are susceptible to both and would benefit from receiving both vaccines. However, little has been known about the safety and effectiveness of combined influenza vaccination. An important study published in the March issue of the IJCP fills this gap in our knowledge. Gasparini and collaborators found that 1 dose of adjuvanted pandemic vaccine is immunogenic in both adult and elderly individuals. In addition, the authors show that prior seasonal vaccination does not interfere with the pandemic vaccine, and in adults, concomitant vaccination does not interfere with the safety and immunogenicity of either vaccine. There are 3 important aspects about the influenza virus biology that we need to better understand. First, we need to understand how to predict superspreaders, which are individuals who infect more susceptible secondary contacts than most others in the population. Superspreaders were described for many infectious diseases, and for many pathogens, it is known that they control most of the transmission events in the population. Second, we need to better understand influenza vaccination in pregnant women, a group at very high risk for morbidity and mortality from influenza infections. Extensive evidence reveals that influenza is more severe and lasts longer in pregnant women, and the risk of complications as well as mortality rates are much higher in this group. Third, We need to learn more about the combined benefits of vaccination and social isolation measures, which are known to save lives during pandemics.